Okay, so first of all, I won't be getting a recording of this on this podium because for some reason, once again, the microphone isn't getting picked up. So we're relying on Chris's recordings whenever they get it. Now, just a reminder that today's lecture is not going to be covered for the midterm or the slideshow component or the practical component this week. So the stuff that we're going to be talking about today, you're responsible for the week after. Okay, so the lab that week, you will need to know this stuff along with the lecture from that week. Question?
Yeah, I'm a little confused with regards to Friday's slideshow exam.
Okay.
So basically, are we going in and then all the microscopes are set up and then a piece of paper, and depending on what piece of paper we're given, that's the answer, like what we look at on our slide and we answer it? And what are we going to other than the first?
So there are two parts. There's a bell ringer and a slideshow. So one will be doing a slideshow, then you move over to a bell ringer. Microscopes, then you're going to be asked questions about the same microscope, and you answer those questions. It's very straightforward.
And then the Saturday exams all here.
All three of those together. So know your stuff for that. Okay? Alright. Okay, so one more question before we get started on blood vessels. Go ahead.
Every single slide component that we looked at, that little like book thing that we use to look at each slide,
Okay.
is it going to be questions from those slides specifically?
Basically, any slide that you guys have in your slide box are fair game.
Okay.
Okay.
But it's just that.
Well, also anything that was on the sideboard is fair game. Anything I showed you in the lecture is fair game. Okay. But basically, any slide that you guys would have looked at in the slide boxes, I have those pictures. And so I can put up any picture I want, and you should be able to figure out what it is. Okay? It might seem a little scary, but if you have been going along and going to other labs, it's really fairly straightforward. You will know the answers within seconds. Other questions? Okay. Let's get started. So today's lecture we'll be talking about blood vessels. So we're departing a little bit from what we've been doing so far. Until now we've been talking about different specific tissues and different cell types. And today we're going to look at how these things are actually put together to form a larger structure. So first things first, I'm going to be mostly focusing today on the blood vascular system, which is what most people think of when they think of the circulatory system. When you think of the circulatory system, you think of the circulation in general in terms of the blood. So you're talking about the blood vascular system, which is subdivided into pulmonary, coronary, and systemic circulation. I'm not going to be making a distinction between them. There are some differences between the systemic or coronary system versus the pulmonary circulation, just based on the fact that the pulmonary circulation doesn't require as much muscle in many cases. And so you might see some differences if you're looking at pulmonary arteries versus other types of arteries. But I'm not going to be making any distinctions there. So I'm just going to be describing arteries in general and veins in general. So what I'll be talking about today is really mostly about general rules for identifying blood vessels. If you go into it in any detail, if you start studying blood vessels on your own as a grad student or something, you might find that things might deviate somewhat from what I described for you. But again, I'm just giving you some general rules for how these things look. Okay? Now, when we talk about the circulatory system, in this case here, I'm not mentioning the lymph-vascular system. There's a lymphatic system as well that carries water and liquids away from your tissues. And I will be talking a little bit about that at the end of today's lecture. Okay? So, for the most part, today's lecture will be focusing on the blood vascular system. And now the function of the blood vascular system is basically to provide a pathway to carry blood around to the other tissues in your body. This allows it to carry oxygen, nutrients to the various tissues in your body. Also allows for thermoregulation, so allows you to heat and cool parts of your body as necessary. Allows you to carry components of the immune system, white blood cells, as part of the blood. And that's going to be carried to all the different tissues of your body as well. So in reality, it is a very, very important component in your body. And depending on what sort of things this type of vessel is dealing with, it will look different. So we will be seeing that different types of blood vessels actually do have a different histological appearance to them, and there are very good reasons for why that is. And so you will be seeing here very much that form will follow function. Okay? So there's a reason why these things are built the way they are. So in general, we can subdivide the blood vascular system into arteries and veins. And so arteries would be defined as the vessels that carry blood away from the heart, and veins would be the ones that carry blood back to the heart. So some people would often say arteries carry oxygenated blood, veins carry deoxygenated blood, but that's not always the case. If you're looking at the pulmonary system, it's actually the opposite. Okay? So in general, these are probably the more accurate way to define these vessels. Now, obviously, the arteries will be carrying blood away from the heart, which means there's a pump there that's going to be pumping blood at a fairly high pressure. And so arteries need to be able to withstand that pressure. So they're going to be specialized to be able to deal with the high pressure that's coming in, especially the ones that are closest to the heart will definitely have to be able to deal with that. And so they are specialized for this. And so they will have more muscle and more elasticity to allow for the smoothing out of the blood flow and to be able to deal with that, the pressure changes that are going to be happening with the pumping of the blood. Whereas the veins tend to be specialized for allowing blood to flow through at relatively low pressure. At that point, the pumping of the blood through the vascular system makes very little difference. It has very little to do with the flow of blood through the veins, through the vascular system there. And so there needs to be another way of getting that blood back up to the heart. In many cases, it's going to be going against gravity. Now, one of the other functions of the veins, or the venous system, is the storage of blood. About 70% of your blood is actually going to be, at any one time, is going to be stored within your veins. So, as I mentioned earlier, blood here is at low pressure, and in many cases will be flowing against gravity, and so veins are also specialized to be able to deal with this. One of the things that happens is that they have valves that will prevent the backflow. Okay. Now, how does the valve work? How many of you have been to the Welland Canal? You should go. It's an interesting sight. So, basically, the Welland Canal and many canals like it are built in a way that, in the same way that our veins are built. So let's say this is a longitudinal section through a vein. We're going to be trying to push blood in this direction. It's just on the inside of the actual lumen. Okay, so blood flow. And so, if it's going against gravity, how is it going to be pumped back to the heart? Well, it's actually kind of a milking action. What's going to happen is that your veins are surrounded by your muscles. And as you move, your muscles compress those veins. And so that means that something is going to be pressing from either side of this vein to make it smaller, generating pressure inside it, and that compresses the blood and forces it forward. Now, it will also force it backwards as well. So, what's going to happen, let's say you've got a little blood going through here, there's compression here that will force the blood going this way and this way. But a majority of the blood is going to be going forward. Why? Because we have valves. These flaps of tissue, this is epithelial tissue, we basically have an open form, which allow for a small opening to be present. And so when you press from both sides here, say you've got these two valves here in place, you compress this region. What's going to happen is that this lumen will become smaller, will become compressed. It forces the blood to go wherever it can. The pressure of the blood will force it through this open valve here. The pressure of the blood downwards will close this valve down below it. Okay? And so majority of the blood will then go up into this chamber up here. When this chamber is compressed, that will close these valves. And again, the blood will be forced upwards again. So, a well in a canal, a very similar system, allows ships to actually go up a hill, which is kind of neat. So I encourage you guys to take a look, at least find out more about it. So that's valves. And again, those valves are basically outgrowths of the epithelium, the endothelium, and a little bit of the connective tissue underneath that will form these flaps. And so again, they will allow the flow of blood to go through them when they're going upwards, but then when the pressure of the blood forces it downwards, they will close to basically a small compartment and allow that pressure to mostly force the blood upwards again towards the heart. So even though arteries and veins have different functions, and they obviously need to be specialized for their specific functions, they do have some things in common. And so what histologists have defined was different layers of the blood vessels. And so what I'm going to be doing today is I'm going to be describing the different blood vessels, the different sizes and different types, in terms of the organization, what sort of things happen within these different layers. Okay? Even though some of those layers may not be clearly visible in many of those cases, we're still just for completeness and for convenience, in many cases, we're going to say that they are still present. It's just that they're very, very thin. Okay? And so blood vessel layers are subdivided into the tunica intima, tunica media, and tunica adventitia. These are basically layers of connective tissue and muscle, as well as some epithelium. So we'll look at them in more detail. But the thing is that these will differ depending on what kind of blood vessel you're looking at. So in order to identify whether you're looking at an artery or a vein, you're going to be looking at the differences between these different layers, and that will help you to identify the type of blood vessel. If you're trying to differentiate between a large artery or a small artery, again, you'll be looking at the different layers of what's going on there to help you identify which type it is. So let's take a look at them in more detail. Here is a schematic diagram that I drew myself, which means it's not terribly good, but that's just my skill with the computer, I guess. And really it's just meant to subdivide these layers for you. So first of all, we have the lumen in the interior of the blood vessel. Then there's a very thin layer of epithelium, which is the endothelium, smooth, simple squamous epithelium that's lining the inside of the blood vessel, which is part of the tunica intima. So the tunica intima is basically this layer here, from this dark line to this dark line on the inside. Okay? So what we have is the endothelium. Underneath that in gray, what we have is the subendothelial connective tissue, which is a very, very thin layer in most cases. It's a relatively loose connective tissue. And beneath that, we have something called the internal elastic membrane, which is symbolized by this darker line. It's relatively thick. I have mentioned this term before to you guys when I talked about elastic fibers, and I showed you an example of it on a slide before, so you'll be seeing that slide again today. This is basically a very thick layer of elastic fibers. In fact, it's like a sheet composed of elastic fibers. Now beneath that tunica intima is the tunica media, which is composed of smooth muscle, and that will be this middle layer here. And so, for the most part, all you find there is just smooth muscle. Now, as you may recall from last week, smooth muscle cells tend to be connected to one another through gap junctions, so that they can communicate with one another to allow them to contract in unison. Also, smooth muscle fibers tend to respond to stretch. So if you want to have a smooth muscle fiber contract, you can try stretching it, and that will cause it to react to that stretch by contracting. Okay? That's something that will become very important when you're looking at blood vessels. And it's also something that's very important when you look at your own digestive system as well. Okay, because there's a lot of smooth muscle there, and one of the ways that it propels things forward is by being stretched first and then contracts around the balls of food. So keep that in mind as you're thinking about, for example, things like arteries and how they respond to the flow of blood from the heart. Now, beneath the tunica media is a layer of connective tissue. This is a relatively dense connective tissue. This is called the tunica adventitia. Now, there is in some cases kind of a boundary between the tunica media and tunica adventitia called the external elastic membrane, which is not quite as obvious sheet of elastic fibers again. Okay, so there might be a tunica adventitia might start at this boundary line composed of some more elastic fibers in a fairly densely packed sheet. Let's take a look at them in a little bit more detail. So the tunica intima is composed of the endothelium, subendothelial connective tissue, and the internal elastic membrane, or internal elastic lamella, that is sometimes referred to. Now this is a very, very thin layer. It is the innermost layer. It is a layer that, in most slides that you'll be looking at, probably will not be very clearly visible. You will be able to see the endothelium, but the subendothelial connective tissue in many cases is far too thin in most of these blood vessels to be able to see it. Part of the reason for that is that it's a fairly thin layer to begin with. But the other part of the reason for that is that most of the blood vessels that we're looking at are taken from fairly young individuals, fairly young animals. And over your lifetime, your tunica intima grows, becomes thicker. Okay, so as we get older, this subendothelial connective tissue accumulates and becomes thicker. What kind of conclusions can we reach based on that alone? You know you've got other layers on the outside of that which can be quite thick. So if you were to think about what sort of consequences does this have, what can you think of if the tunica intima becomes thicker with age?
The more thicker, the less elasticity we can have.
Okay, so the thicker it is, the less elasticity it might have. Okay. It's not really what I'm looking for. It's a possibility, I guess. Yeah.
The thicker it is, the more difficult it is for materials to diffuse across.
The thicker it is, the more difficult it is for materials to diffuse across. Okay, that's a good thought. If you think about this, if we have the lumen, here's our endothelium. And let me draw the internal elastic membrane, but not with this marker, because that's invisible. What you'll find is in most slides that internal elastic membrane is actually quite wavy. So here is our subendothelial connective tissue. And the internal elastic membrane. Okay, so you think, okay, if we have an increase in thickness, we're going to lose some of the ability of this tissue to allow things to diffuse across. Because again, from the interior, we're going to have things like oxygen and nutrients diffusing across. Okay? But really, what determines the ability of the tissue to allow things to diffuse across? Yeah.
Since it maybe like it gets narrower, so you have less blood flow.
Okay, so you're getting to where I want to, but what determines the tissue's ability to allow diffusion? How much water there is, right? So the thickening of this tissue doesn't necessarily mean anything in terms of the amount of water that it contains. And so the diffusion probably won't be affected as much. Okay? But you do bring up a good point. If you think about this, if we take this and we look at it when it's older, if we have, again, the same elastic membrane, internal elastic membrane, same size, but we have a lot more of this internal elastic, this subendothelial connective tissue, and it is thicker, what's going to happen is that our lumen becomes smaller, doesn't it? So here's our endothelium now. Okay, so let's take it a step further. So if our endothelium is now smaller, our lumen is smaller, what does that mean?
Is it like increasing pressure?
Thank you. So it increases blood pressure. Okay? So you're still pumping the same volume of blood with each heartbeat. So the volume of blood doesn't change, but now you're pumping it through a smaller opening. So the pressure inside that blood vessel increases. Okay? So this is older blood vessel. So what we're really seeing here is going to be hypertension, which is relatively common in older individuals. It's a perfectly natural thing. Now, what normally do we also seem to associate with older individuals? What sort of other problems with vascular system do we normally associate with? Yeah,
Heart attacks?
heart attacks. Okay.
I'm just thinking like
Okay.
clogging of like.
Okay, that's something different. We're kind of jumping ahead of me there, but we'll get there. We'll get there. Okay. Yep. Hardening of blood vessels. Okay. Yep.
Like plaque on the inside?
Right. Atherosclerosis. Oops. Go off the screen now. You guys gotta warn me when I go off the screen. Okay. So atherosclerosis. And this idea of hardening of blood vessels. If they're hardening, they're less likely to expand and contract again. So they're becoming less elastic as well. And that's because of the buildup of plaques, which is usually calcium. So there's calcium buildup within the tissue. That happens within that subendothelial connective tissue. That tends to happen because of the buildup also of cholesterol. Okay. So we have a narrowing of blood vessels, which is going to increase blood pressure inside of the blood vessels. Something that our endothelium wasn't necessarily designed to deal with initially. And so our endothelium will become damaged at some point. So, first of all, let's take a look at this blood vessel now, this older blood vessel in the longitudinal section. So let's draw the longitudinal section of this blood vessel. Here's our subendothelial connective tissue, this collagen. And let's put in the internal elastic membrane. So we have blood flowing through this. Now, at some point, again, this blood is flowing at higher pressure, so we have increased pressure and also increased vascular resistance. So basically there's more friction against that surface. Now, one of the functions of the endothelium is to provide a nice frictionless surface for things to flow past. But when you start causing damage to this by having this increased flow increased pressure, you're going to start damaging the endothelial cells. And so you might eventually get one of these edges to get turned a little bit. And that tends to mean that we're going to introduce turbulence into the flow of blood. And so instead of blood flowing as a nice straight flow, what's going to happen is that there's going to be a little bit of a bump here, and that's going to mean that some of the blood, some of the current will be disrupted. You can think of as basically the flow of water in a stream. The bottom of that stream is not going to be very clean if it has a rough bottom, because it's going to start to collect, because it starts to have turbulence at the bottom, and that means the collection of things. In this case, it's the collection of things like platelets and clotting factors. Now, under normal conditions, your clotting factors inside your bloodstream are at concentrations that are not likely to cause any problems. But it's actually fairly easy to start the cascade that begins the process of clotting. Okay? And so eventually what's going to happen is that we're going to have an increased amount of these clotting factors showing up and just kind of building up and behind one of these little upturned edges of the endothelial cell or the damaged cell. And so we end up starting to build up a little bit of a clot. And as more blood flows past it and it becomes bigger, it generates more turbulence. That clot gets bigger and bigger and bigger and bigger. Now, this clot could do one of two different things. It could stay there and keep on becoming bigger and bigger until it includes the whole blood vessel. Okay? And then you have no more blood flow passing through it, and so all the tissues beyond this point are deprived of oxygen. So in this way, you're not getting a heart attack. It's not quite that likely in this case, but you could. Okay? Or you could take this, which is referred to as a thrombus, while it's still attached to the endothelium. And that thrombus might break off. And so you might have this breaking off to produce an embolus. So this clot, this freed clot, can now pass through the vascular system. What do you think is going to happen to it?
You'll either get a stroke or a pulmonary embolism.
Okay. A stroke or pulmonary embolism, right? Yes. Now, pulmonary embolism is more likely to happen if you actually have one of these things building up in the venous system. We talk about that shortly. In the arterial system, if this is an artery, what's going to happen? What do you think? Yeah.
It's going to get clogged in the arterioles or
So it's going to keep on flowing through the blood vessels until it's too large to pass through one of them. And so it's going to get clogged in one of the smaller arterioles or a capillary cell. And so, again, it could lead to an embolism that could be like that, something that could cause a stroke or something that could cause a heart attack. Okay? Again, with pulmonary emboli, usually the origin of those is going to be within the venous system. And that's because it's going through larger and larger and larger veins until it gets to the heart and it gets pumped into the system of the lungs, and it gets lodged there somewhere. Okay? But if it's something that starts out in an artery to begin with, chances are it'll end up in some sort of a small capillary bed somewhere else. Okay, so that's the tunica intima. Let's get back to the slides. So we've got that endothelium. Again, that endothelium seems like a fairly boring sort of structure. It's just a smooth surface. But it can actually have a very, very important function in many things, including clotting, including the formation of these thrombus or thrombi or the emboli. But also, in the case of just regular damage, let's say you cut yourself, okay, or you scraped your knee or something and you start bleeding. you've just damaged the endothelium in a blood vessel. And what's going to happen at that point is your endothelium becomes activated. Okay? Under normal conditions, your endothelium is designed to prevent clotting. But when your endothelium becomes activated, it actually changes and it becomes pro-clotting. Okay? So it starts the whole process of clotting because it senses that it's damaged, and one of the things that you want to do is to stop the flow of blood, just so that you're not losing blood. Okay? Also, one of the things that happens with the clots is that they tend to contain things like growth factors, which will cause the migration of cells into the area so that they can actually rebuild the damaged tissue. Okay? So in cases where the endothelium is actually damaged in some way, it will become what is known as activated, and at that point it actually changes its characteristics very much and becomes very much all about starting the clotting process. It becomes very sticky and starts to send up all kinds of chemical signals to other cells to bring them into the area to start things like the inflammatory process, which we'll be talking about in the coming weeks. Okay? So it might seem like a very boring kind of tissue, but actually is very, very interesting. It actually has a lot of different things that it can do. Again, underneath that is that this connective tissue composed of collagen. There might be some fibroblasts in there, and also macrophages, and also smooth muscle fibers. The Tunica Media is composed of smooth muscle, and some of those muscle fibers can actually migrate into the tunica intima through the internal elastic membrane. Internal elastic membrane is a fairly continuous sheet, but has fairly large pores to allow for diffusion to occur. And so smooth muscle fibers can actually migrate into the region. And a lot of this material here, the collagen and elastic fibers, are actually produced by those smooth muscle fibers themselves. Now, I want to introduce another term here, and that is the term foam cell. What is a foam cell? A foam cell is basically either a macrophage or a smooth muscle fiber that becomes filled in with with droplets of fat. Okay? So things like if you have a high cholesterol diet, that cholesterol may become deposited within this endothelial connective tissue. It is not something that should be staying there. And so things like macrophages will come along and try to take that up. They're going to accumulate it, and so you end up with a cell that is filled with these little droplets. And so that macrophage, after a while, will look like this. This is your macrophage. Then is going to have enveloped a whole bunch of these lipid droplets and just become this foamy looking cell. So it's called a foam cell. The same thing can happen to those smooth muscle fibers as well. They migrate into the area, they take up that lipid to try to sequester it away, and they end up just becoming filled with these small droplets of lipid. And so again, they will also be referred to as foam cells by pathologists. Okay. So what happens? Well, you have an accumulation of this stuff here, so let's just use this diagram still. Okay. So let's say we start to accumulate these foam cells here. So there's going to be a lot more material in this region. And so what do we see? We see a bump on the surface of this blood vessel, which causes a narrowing in that blood vessel, causes a disturbance in the flow of blood. Okay. It's going to cause turbulence, which means what? Sorry? Uneven flow of blood. Okay, so we've got turbulence showing up here again. So what's going to be the next thing that's going to happen? Accumulation of clotting factors, a blood clot forming, etc., etc., etc. So again, atherosclerosis ends up causing the same sort of thing. Okay. And again, usually underneath one of these bulges, you will find a lot of these foam cells. In many cases, they will become calcified over time. And so actually under a microscope, they tend to actually show very intensely stained, just very purple staining cells. Okay, so again, internal elastic membrane, I already told you about that one. Just a fairly continuous sheet of elastic fibers that has pores within it. So it's a fenestrated sheet, which allows for passage of things like smooth muscle fibers. It also allows for diffusion of things from the lumen through that subendothelial connective tissue into the underlying layers. Question?
Did you say it seems dark?
Stains fairly purple. So wherever you have calcification, you saw calcification on your slides for bone. It was very purple material,
But
right?
the fat in itself is...
The fat itself won't stain, but once it accumulates like calcium, you tend to see these streaks of purple showing up in these regions. Okay, so underneath that tunica media, the tunica media, this is the middle layer, the muscular layer, contains smooth muscle. And so the smooth muscle here is responsible for the production of the ECM. So the reticular fibers that are going to be found within the ECM here, the elastic fibers that are going to be found here, the type 1 collagen that's going to be found here, is going to be produced by the smooth muscle fibers. Now, it is made up of smooth muscle in arteries. If you look at a vein, chances are what you'll notice is that in this layer, the layers of smooth muscle are actually a little more separated from each other. And you might notice some smaller nuclei, which probably belong to fibroblasts. So that one of the ways to differentiate the two is really to look for how tightly packed are the smooth muscle fibers. If they are tightly packed in all layers, that's probably an artery. If you're looking at one that looks like you've got a fairly large amount of ECM in between layers of smooth muscle, and you might see some of the smaller nuclei that might belong to fibroblasts, you're probably looking at a vein. Now, it is quite thick. In arteries, this is the thickest layer of the blood vessel. Okay? So in arteries, you can have up to about 40 layers of smooth muscle. So something like your aorta is going to have a lot of smooth muscle. And as the blood vessels get smaller, they get fewer and fewer layers of smooth muscle. And so again, if we're trying to differentiate between a large blood vessel, a large artery, and a small artery, what you want to do is just take a look at how many layers of smooth muscle do you see in that tunica media. Now, in larger arteries, you will often see an external elastic membrane or an external elastic lamella, which again is a fairly continuous, not quite as obvious, a relatively continuous sheet of elastic fibers. And again, it's a fairly fenestrated sheet. But again, it's usually not as obvious as the internal elastic membrane. The other things that we tend to see in large arteries and large vessels in general, so arteries and veins usually, especially closer to the heart, is we tend to see elastic fibers, fairly prominent elastic fibers within the tunica media as well. And so again, they can be fairly long, continuous sheets in between the layers. Whereas here, the pressure inside the chamber of the heart is lower. This is inside heart. Now, being science geeks, let's try to graph this. And so we're going to look at pressure. And then at the bottom, let's look at distance from heart. So initially, closest to the heart, the blood vessels that are closest to the heart, during systole, they will receive very high pressure, and then diastole, low pressure. Okay. And then high pressure again further away from the heart, because again, blood flows as one big amount of blood, and then nothing. And then one big amount of blood, and then nothing. Okay. So as you go through that blood vessel, the further you get from the heart, you start to see kind of a weakening in the amount of pressure that is actually detected within the blood vessels. Okay, so what we're looking at here is, this is in blood vessels, in arteries. visible. Different color. So what we have is basically this high pressure followed by low pressure, high pressure, low pressure. But the further away you get from the heart, what happens is it just starts to even out. And so eventually what you have is a fairly even flow of blood passing through these blood vessels. Why is that? It's because of the fact that we have a very thick tunica media and a fairly large amount of elastic fibers initially. So recall that smooth muscle responds to stretch by contracting. So as soon as this high pressure hits that aorta, how is it going to react? It's going to react by contracting. So initially that blood flow is just going to stretch it, and it's going to react to that by contracting back. Okay? What's going to happen at that point? Well, that contraction is going to send that blood forward. Also, it's going to be sending it back. Sending it back towards the heart is going to close the valves in the heart, so that it can refill its chambers again. So it's an important thing as well. But as that bolus of blood is flowing through, again, it's going initially at high pressure, but as it's constantly being reacted to, it smooths out and makes that pressure smaller and smaller. And that differential between high pressure and low pressure becomes smaller and smaller over time as you're going further and further away from the heart, until once you get to your arterioles and your capillaries, you're dealing with a relatively small differential between the systolic and diastolic. And so at that point, you don't really have much of a difference in terms of how the blood is flowing. And so these smaller blood vessels don't require to have that large amount of muscle ability to respond to these changes in pressure. So you're not going to see as much of the elastic fibers there anymore, and you're not going to see as much muscle there anymore either. It's not really required anymore. And so again, when you're looking at blood vessels under the microscope, if you see one that has lots of smooth muscle, you're probably looking at one that deals with a lot of these changes in pressure, and has to try to smooth that out. If you're looking at an artery and you're looking at a lot of smooth muscle fibers in that tunica media, and you're seeing a lot of elastic fibers, you're probably looking at something that's close to this region right here, very, very close to the heart, like the aorta. Okay? And some of those initial blood vessels that branch off from the aorta, all of those will have a lot of that elastic fiber within the tunica media to really help with the recoil back to its original shape. Okay. Now, conversely, if you were to think about this as well and think, what about veins? Would they require a lot of smooth muscle in the tunica media? No, they're not dealing with the same amount of pressure. They're dealing with very low pressure, in fact. Okay? So having a very thick, dense wall would actually be a problem for veins, because what you need to have with veins is the ability to collapse in, to be compressed by outside sources. Okay? So there you don't want to have lots of smooth muscle. You do not want to have a very thick wall. Okay. The outermost layer is the adventitia, tunica adventitia. And so here we're looking at a fairly dense, irregular connective tissue. So you guys hopefully remember what that looks like. Lots of fibroblasts. There's going to be some macrophages there, mast cells, things like that. Now, this is the layer that is thickest in veins. So arteries don't really have a very thick tunica adventitia. It's still there, but it's relatively thin compared to everything else in that artery, especially the tunica media. Whereas in veins, this is the thickest layer. Why? It gives it some structure. Okay? So this is the part that really gives the vein their structure. Okay? Basically forms the wall of that particular vessel. Now, the tunica adventitia is where we tend to see something called the vasa vasorum, which is the vessel of a vessel. It's a blood vessel that feeds blood vessel. Which seems a little counterintuitive. It's already a blood vessel. Why does it need its own blood vessel? Why does it need to be vascularized? Well, if you're a vein, in most cases you are dealing with blood that is low in oxygen. In some cases you will be also dealing with blood that is low in nutrients. If you want to feed your cells, you're going to need to get that from someplace else. So especially in the veins, we tend to see a fairly prominent vasa vasorum within the tunica adventitia. Because, again, if there's going to be any diffusion from that lumen, it's not going to reach very far because there's not much there to begin with. There's not much oxygen to be taken out of that lumen at that point. It's fairly oxygen poor in most cases. Okay? So you need to be able to feed that blood vessel in some other way, especially the larger blood vessels that have a lot more structure to them. And that's where we tend to see that vasa vasorum showing up. They may also contain a nerve supply. Blood vessels in general have a nerve supply to some extent. Now, blood vessels, as I mentioned earlier, if you want to contract a blood vessel, you just need to stretch it. So smooth muscle will respond to stretch. But sometimes you do want to have your blood vessels constrict without stretching them. Any examples that you can think of? Or you might want to dilate your blood vessels without doing anything to them. So you want to have some sort of control over the size of the lumen. Can you think of any examples when the size of the lumen of your blood vessels might change without you actually pushing blood through it? Yeah.
For temperature control?
Temperature control. Okay. Good. Temperature control. So we talked a little bit about this last time. For example, if you have a muscle that's been working very hard for a while, all of a sudden the skin around it feels very warm. Why? Because a lot of blood just flowed into that region, and then it's trying to cool off by sending that blood close to the surface of the skin. So that means that there are going to be small arterioles which are going to open up to allow blood to flow through. Okay, so there's going to be some control over the size of the opening of those blood vessels. How about the other way? You're outside in the middle of winter and it's minus 30 degrees outside. You try not to go outside at that point, but if you have to be, what's going to happen? Especially around your extremities. Your skin gets very cold. Blood vessels there constrict to slow down the flow of blood into those regions so that you keep more of the warm blood around your core to keep your core warm, because your extremities are expendable as far as your body is concerned. It's okay if you lose an arm, but you can't lose anything in your core. Okay? So again, that's a way of controlling the flow of blood without actually having to expand or doing anything to the smooth muscle in some physical way. There's got to be another way of controlling it. So again, that nerve vascularis, the free nerve endings that are reaching it, would be one of the ways that this can be controlled. Smooth muscle can also respond to hormones as well. Became some hormonal control. So for example, things like, you might think of one or two signal molecules that might affect your blood pumping. Things like maybe epinephrine, adrenaline, right? So there's ways of controlling the flow of blood through your arteries and veins by controlling the contraction of that smooth muscle in the tunica media, and in some cases that goes through stretch. In some cases, that happens through innervation. In some cases, it's just a signaling molecule that's sent out throughout the body. So let's talk about arteries and veins and try to differentiate between them. Let me just make sure that I haven't missed anything important. Okay, so we want to look at arteries and try to compare what kind of artery we're looking at. Well, first of all, I want to remember that classification is based on the differences between the layers and what the layers look like. Okay? So when you're looking at arteries, which layer do you think you're going to be looking at? Tunica media. Right? That's the thickest layer, the most obvious layer in arteries. And so that's the one that we will be really looking at to try to differentiate between them. Now, obviously, arteries, large versus small, differ from each other based on the size of the lumen as well. Okay? But under the microscope, you can't really measure the lumen very easily and say, oh, this is a small lumen, this is a large lumen, unless you can actually see the whole lumen doesn't fit, or the whole blood vessel doesn't fit on the slide, as opposed to this one where it does fit on the slide. Okay? So in many cases, you're left really looking at the tunica media to give you some idea of what type of vessel you're looking at. So let me give you some guidelines. If you're looking at large arteries, so especially something like the aorta, these are generally elastic arteries. If I say elastic arteries, that means that we're looking at layers of elastic fibers within the tunica media. So we're not just looking at the internal elastic membrane being present, but also you're going to have many other layers of elastic fibers in between the layers of smooth muscle. So the elastin there is very, very prominent in the tunica media. If you see that, you're looking at a large artery. If it's elastic, it's got to be large. It's going to be one of those, the aorta, or one of the initial branches off of the aorta that you would normally see. Now, in terms of diameter, usually the diameter here is more than 1 centimeter for the lumen itself, not the diameter of the blood vessel, but the lumen diameter is about 1 centimeter or more. Now, the next one down is a muscular artery. So once you start to see that the tunica media no longer contains a lot of elastin, you might see still a couple of strands or a couple of sheets showing up here and there. But if the majority of that tunica media looks like it's just smooth muscle, you're looking at a muscular artery. And so at that point, we're further away from the heart. Okay? So it's going to be a smaller artery. Okay? So in those cases, what we're looking at is a diameter that is about 2 to about 10 millimeters. So less than 1 centimeter, about 2 millimeters to about 1 centimeter. And again, you can't really tell that under the microscope because that's not going to fit under the microscope. Okay? It's not going to fit on the slide. So what you're looking at number of layers of muscle. So first of all, can you see any elastic fibers within this? If you're seeing lots of elastic fibers, it's probably an elastic artery, it's probably a large one. But if you don't see much elastin there anymore, you can probably fairly safely say it's probably a muscular artery, so therefore probably a medium artery. Okay? In terms of the number of layers, up here we're looking at about 50 layers of smooth muscle. Again, don't count every single one, estimate. Okay? If you're looking at a medium-sized artery, we're looking at something close to about 10 to 15 layers of smooth muscle in the tunica media. Okay? So a fairly substantial amount of muscle still, but nowhere near what we were seeing initially closer to the heart. And then again, as we progress through, we get progressively smaller and thinner vessels. And so with a small artery, we're looking at a diameter of close to about 2 millimeters or less, so about 0.1 to 2 millimeters. And there we're looking at a tunica media that has maybe between 6 or 7, maybe 8 to 10 layers of smooth muscle. And then we get to arterioles. These are the smallest of the arteries. And throughout all of this, you are still going to be seeing an internal elastic membrane, a fairly prominent internal elastic membrane, even all the way down into the arterioles. They still have a very prominent internal elastic membrane. Okay. But by the time we get to the arterioles, we're looking at a diameter of about 10 to maybe 100 micrometers for the lumen. And we're looking at maybe 1 to 2, maybe 3 cell layers of smooth muscle in the tunica media. By the time you get to this point here, the arterioles, that tunica intima is going to be very difficult to see. You don't see that subendothelial connective tissue anymore. You do see that endothelium, and you do see the internal elastic membrane, but you don't really see much in between them. Okay? So there might be a few fibers there, but there really isn't much thickness to these things. In fact, if you're looking for a tunica intima, and if you're looking for that subendothelial connective tissue, you're most likely to see it in the large and medium-sized arteries. By the time you get to small arteries and arterioles, you usually don't see that internal elastic membrane, not the internal elastic membrane, so the subendothelial connective tissue. That internal elastic membrane that will be visible all the way down to the arterioles. Now, the arterioles are, in many cases, or at least in some cases, referred to as the precapillary sphincters. So, precapillary sphincters. And that's because that smooth muscle in the tunica media there tends to control the flow of blood into the capillaries that follows the arteriole. And so the arteriole will control whether anything actually does go through or if nothing goes through, or if only some blood is going to be allowed to pass through it. Okay, so This is where that control of the flow of blood into a region is actually going to be occurring. Now, after this, blood will flow into a capillary bed. We'll talk about capillaries very shortly. And then after that, it will enter the venous system. And the first vessel that will be entering is the venule. Okay? And so venules come in two sizes or two types. There's just a regular venule and a post-capillary venule. So let's do that there as well. Post-capillary. Please do keep that in mind because that distinction will be important. Post-capillary venules tend to have a diameter of between 10 to 50 micrometers and no muscle. So you can just think of them as very large, very wide capillaries. And then the regular venules have a diameter of about 50 to 100 micrometers. They might have one or two layers of smooth muscle in the tunica media. Question?
So for the arterial, you have the capillaries after the arterial, and then sphincters within the capillary?
So the sphincters are, the arterial is called a precapillary sphincter. Okay? The sphincter itself is basically the smooth muscle. So the smooth muscle of the arterial will control the flow of blood into the capillary bed.
So it acts.
So it acts as a sphincter, yeah. So you can think of it, just try to picture it this way. You have an artery setting up an arteriole, and that opens up into a capillary bed. Okay. And those capillaries then collect into post-capillary venules, into larger venules, and then the venous system, which are vein. Okay. So this is a vein. Artery. This is arteriole. You may want to leave some space on this diagram. We'll be adding to it later, since you already have one. And so what we have here is a capillary bed. And so the smooth muscle found within this arteriole can constrict it completely and basically close it off so that there's no blood flowing into this capillary bed. Like, for example, when you're sitting right now, and so your thigh muscle isn't really working very hard. So chances are not all the capillary beds need to be working 100%. And so not all of them are going to be filled. So you're going to have a lot of these arterioles, these precapillary sphincters, closing off so that you're not wasting resources pumping blood through all these capillaries and all of these capillary beds. So again, initially we have relatively small venules that will collect the blood from these capillary beds. Initially, those post-capillary venules do not have any smooth muscle in the tunica media. That will become important when we talk about things like inflammation next week, or not next week, 2 weeks from now. And then, again, it will flow into small veins. Small veins are a little bit larger. They have a tunica media with about 2 to 3 cells, two to three smooth muscle cell layers. And the diameter there's about between 0.1 millimeters to about 1 millimeter. Then into a medium vein, diameter there anywhere between 1 and 10 millimeters, and about 3 to 5 layers of smooth muscle. And then we get into the large veins, especially the largest ones would be elastic, especially the vena cava. And so here we're looking at a vein that is, or a vessel that's going to be larger than 1 centimeter in diameter. And is going to have about anywhere between 5 and 15 layers of smooth muscle. Okay, so again, a very thin layer in that tunica media. Now, when you're looking at these blood vessels under the microscope, one of the things you'll notice is that they will kind of look, in general, at very low magnification, kind of like this. Between these two, this would be the artery. This would be the vein. The arteries tend to, under the microscope, look very round and open. One of the reasons for that is they have a very thick wall that holds them open. Okay, so even after slide preparation, an artery will look nice and round. The vein tends to collapse during slide preparation. When we cut it, it just kind of tends to scrunch in because it doesn't have much structure to it. Compared to the size of the lumen, that wall is very, very thin. And that's one of the things that allows it to be compressed by your muscles to allow that blood flow to go back up to your heart. So you want them to be compressible easily. So that wall isn't going to be very thin. The other thing I want you to notice is that the lumen of the artery is much, much smaller than the lumen of the vein. And last thing is that they tend to go together. So usually if you see an artery, there's probably a vein very close nearby. In fact, that association can be so close that when you're looking at the venules and arterioles, usually they will actually share the tunica adventitia. So they're used to usually right side by side next to one another, and the tunica adventitia of the artery, of the arteriole, merges with the tunica adventitia of the venule. Okay, let's look at some pictures. We'll get to printability soon. Okay, so the next few slides, I tried to make sure that I got them at about the same magnification, using about the same conditions for both the vena cava and the aorta. So what you're seeing on this slide here is a fairly low power micrograph of the section through an aorta, so cross section. On this slide, on the right-hand side, is the lumen. So the endothelium would be right at the edge here. If you look very closely on your own pictures and on your own computers, there are a couple of red blood cells showing up right at the very edge of the endothelium, so you can see just how small they are compared to everything else. So you're looking at a very large vessel. Okay? The reason I point out red blood cells is that red blood cells are often referred to as the microscopist ruler. They have a very specific size, and so if you can find red blood cells on your slide, you can measure anything you want. Okay? The diameter of a red blood cell at typical slide preparation like this is about 6.5 to 7 micrometers in diameter. Okay? So that tiny little dot right there and there, which if you're sitting back here looking at this stuff probably doesn't look like anything, but if you have a picture in front of you, you might be able to see it, is about 7 micrometers. Okay? So this is a heck of a lot thicker than a few micrometers. Okay? Now, what you can see here is from here to about here is the tunica intima. And I don't expect you to take my word for it. We will zoom in a moment to take the next slide. We'll look at it in a little more detail. But basically, from here all the way to here is the tunica media. So right there is where the tunica media ends. And then to the left of that, all of this down here, it's a little bit damaged. That's just slide preparation. That's just an artifact of slide preparation. Okay. But all this back here is the tunica adventitia. Okay. In fact, out here, if you look carefully, you'll notice there's a blood vessel right there. That would be the vasa vasorum. Okay. It's kind of the tunica adventitia. And again, because of how thick this blood vessel is, it does require its own vasculature. Okay. So we can clearly see that even in the aorta, you're going to have this going through. Now, the aorta is carrying oxygen-rich blood and nutrient-rich blood, hopefully. But chances are it's passing by way too fast for any infusion to really occur very efficiently. So, again, you're going to need to feed these tissues somehow. Let's zoom in a little bit. So this is a high magnification of that portion that is closest to the lumen. So again, we can see the lumen here. Here's the red blood cell, right there. We have the endothelium right at the very surface. This squiggly line that you can kind of make out here would be the internal elastic membrane. So everything between here and the endothelium is a subendothelial connective tissue. So you can see here it's relatively large, relatively thick in this particular blood vessel. But again, we're looking at a very large blood vessel. As we get into smaller and smaller blood vessel, that subendothelial connective tissue becomes thinner and thinner. Then beyond this internal elastic membrane, you see multiple layers of what look like more of the same sort of thing, more of the same kind of elastic, sort of wavy-looking lamellae. Okay? That's because this is an elastic vessel. So what we're seeing here is multiple layers of these elastic sheets. Okay? So in this case here, that internal elastic membrane is not terribly prominent because what follows it is more of the same sort of thing, and interspersed with layers of smooth muscle. Now, smooth muscle is what's responsible for producing those sheets of elastic fibers. So what we have here are these nuclei. These nuclei belong to smooth muscle fibers. Then you have a layer of elastic fiber. There's another layer of elastic fibers over here. What's in between them is the smooth muscle itself. Okay? And again, if you were to try to count all of these layers of smooth muscle, you'd come up with a lot, a very big number. Okay? Let's not try to count these things. Here's the vena cava for comparison. So again, we're starting out at low magnification, so let's flip back a couple of slides. Here's the aorta. Okay. Notice how thick it is. This is going to be the same magnification now for the vena cava. Okay. Much thinner. Okay. Again, that wall doesn't have to be as thick. Okay. It doesn't need to be. It's not dealing with very high pressures at this point. Okay. Again, on the right-hand side is the lumen. And then we have from here to about here, and I also kind of make out as kind of a squiggly line going across from the top to the bottom. It's not very continuous, and that's one of the things about the internal elastic membrane in veins. If you do find it, usually it's not as obvious and it's not as continuous as it is in arteries and arterioles. So this is the internal elastic membrane. Everything between that internal elastic membrane and the endothelium over here is the subendothelial connective tissue. Now, here's the weird thing. This is going to be difficult for me to just show you with a pointer that shakes. So, let me try to get up here. Hopefully it doesn't collapse under me. From here to here is the tunica media. Right there. That's the tunica media right there. Okay? Yeah, it's not very thick. Everything from this point to the left is a tunica adventitia. Okay? From this point to the left is a tunica adventitia. Okay, let me draw it down now without breaking anything. You may not believe me. Let's zoom in. Again, lumen up here, endothelium, internal elastic membrane right there. It's kind of a wavy band that you're seeing here, some of this continuous. Okay. So subendothelial connective tissues right here. Then from here to here is the tunica media. How many layers of smooth muscle can you count there? That should be easy enough to count. Smooth muscle fiber, smooth muscle fiber, smooth muscle fiber, smooth muscle fiber. How many layers? 4 or 5? Yeah. Okay. Not very thick. But wait a minute. What does this look like? back to last time. Something somewhat unexpected, based on what I've told you so far. This region right here is full of cells. If it's very cellular, its chances are it's not connective tissue, right? You know, connective tissues are fairly acellular, so it's not connective tissue. What are we looking at there? Obviously, it's not epithelium, there's no surface. What do you think? Cartilage? What are we looking at? Boy, smart guys, you're studying this stuff, right? Because you've got a test coming up. Yeah. Cartilage. No, it doesn't really look very basophilic in the ECM. And there's, again, cartilage is a connective tissue, so it would be mostly ECM, right? So this is mostly cells. What are we looking at?
Looks a bit like skeletal muscle.
Looks a bit like skeletal muscle, okay? A bit, maybe.
Bone marrow.
Sorry? Bone marrow. No. We don't see any bone around there. We wouldn't expect any bone around there. This is the vena cava. So you're closest to the mark. It is muscle. We know it's not epithelium. We know what epithelium looks like. There's no free surface. We know it's not connective tissue. It's too cellular to connective tissue. That leaves muscle, right? So what kind of muscle? It might look kind of like skeletal muscle, but again, you might be thinking about the wrong scale. Okay. And remember about skeletal muscle, you can control it, right? You can control the muscle around your vena cava. Okay. What else could it be? Smooth muscle. Was that a question or an answer? Yes, it's smooth muscle. Okay. It's not cardiac, obviously, because there's only one place we're going to find cardiac. I didn't say anything about vena cava when I was talking about cardiac muscle. So, smooth muscle. It's a cross section. That's why it looks a little weird. Okay. What does a cross section of smooth muscle look like? And why? Remember, we've got a smooth muscle fiber, this very long muscle fiber. And here's the nucleus. Okay. What is it going to look like if we cut it here? It's going to be round, cross section, and it's going to have a nucleus in the middle, right? What about if we cut it here? It will look round. Is it going to have a nucleus? How about here? How about here? How about here? Any of those? So if you're looking at smooth muscle in cross-section, what you tend to find is you have varying diameters of relatively round-looking cells. And some of them have nuclei and many of them don't. It's just because of how long the fibers are. Your chances of cutting right across a nucleus are relatively low. Okay? Okay, so let's take a look at our slide again. What are we seeing here? Well, here's around cell and nucleus. There's cell without a nucleus, cell without a nucleus. So you're seeing a whole bunch of rounded cells that don't have nuclei, and there's a few that do. So we're seeing bundles of smooth muscle in the tunica adventitia. I want you to remember this. It will show up on the exam. Not this coming one, but the next one. I am almost sure of it. Okay. One of the things that distinguishes the vena cava from all other veins is that you have all these layers. So you have this endothelial tissue. You have your smooth muscle layer, so you've got your longitudinal sections of smooth muscle that go around the outside. Okay. There's not a lot of them. And then what you have are bundles of smooth muscle cross section. Those bundles are going to be separated from each other by the different tissues, so that's what you're seeing kind of showing up in orange in that particular slide. You just have these individual small clusters of these cells, and so from here to here is your adventitia. If you're trying to picture this, let's pretend this is the tunica adventitia. This is the vena cava. Okay, so going up and down. Okay? The smooth muscle in the tunica adventitia would be organized like this. It would be running along the length, parallel to the length of the vena cava. Okay? So in most blood vessels, you have got a tunica media, and that's where the muscle is. And so if we were to take a look at a cross section through it, you have your smooth muscle arranged like this. So all the muscle fibers will be arranged circumferentially around it. But in the tunica adventitia of the vena cava, you also have smooth muscle arranged longitudinally along the length of the vena cava as well. Can anybody think of a reason why that might be something the vena cava might want to have? Yeah. Provides more strength. Okay. Sorry? Okay. Okay, so you're going to have more muscle, so it's going to have a little bit more thickness to that, more structure to that tunica adventitia. I'm not sure that the strength is really what the vena cava would be looking for at that point. Yeah.
The axial contraction of the muscles that belong to the leg.
Okay, sorry. One more time.
If the muscles are arranged along the length,
Mm-hmm.
like they all contract if you like the last leg.
Okay, that's what we're looking for. Okay, so if you think about this, how many of you are anatomy students or took anatomy and did dissections? Don't be shy. Don't be asking too many questions. So when you did your dissection, if you looked at your vena cava, was it surrounded by lots of muscle? I mean, most of the veins, for example, veins in your legs are passing through. They've got lots of skeletal muscle around them to compress them. What about the vena cava? Does it have lots of skeletal muscle around it? Again, remember, something's got to push that blood towards the heart. Vena cava doesn't have anything around it. It's going through the abdominal cavity. There's organs around it, but nothing specific that would compress the vena cava. So in this case here, what we have are these bundles of smooth muscle around the outside. And what happens to smooth muscle when it contracts? You go from a cell that looks like this to a cell that looks like it shortens, but the volume is still the same, so it thickens. Right? So if you think about a whole bunch of these cells doing the same thing, it's going to compress the lumen. So this is why vena cava is going to have that, whereas most other veins will not. You're not going to see a lot of smooth muscle in the tunica adventitia and other veins or blood vessels in general, but in the vena cava, that is the characteristic feature. Write that down. Memorize that because it will probably show up at some point. Okay, moving along. New slide. What do we see here? We've got artery and vein. Which one's the artery, which one's the vein? Based on what you know. Which one's the artery, which one's the vein? Yeah. Artery on the right, vein on the left. Why?
The lumen is still maintaining a circular shape.
Okay, so it's a nice round lumen.
The thickness around
The thickness around the artery, thickness of what? Another terminology. I gotta stop thinking over here. People will stop wanting to answer questions. Okay. So the Tunica Media is quite thick. And I'm glad she did this, okay? Because you don't want to just go with one piece of evidence. The lumen is round. So what? Maybe it's just a weird slide. So look for more than just one piece of evidence for why you think it is what it is. Okay? So you think it's an artery because you have a round lumen, whereas this one's very collapsed. What else might tell you something about the kind of vessel you're looking at? Well, you've got a Tunica Media here that is very thick compared to over here, which is probably very, very thin. You barely just make out the Tunica Media here. Okay? So again, another piece of evidence. What else? Notice this very dark, wavy, squiggly line at the very surface. That's the internal elastic membrane, very clearly visible in the artery. You can look around at the vein. There's a slight bit of darker staining in that region close to the lumen, but it's nowhere near as obvious as it is in the artery. Okay? So obviously the internal elastic membrane here is much thicker, much more prominent. And again, you can see that very clearly within the artery. Okay? Again, smaller lumen, larger lumen. Okay? If you were to try to guess, what kind of artery would this be? Small, medium, large? So keep in mind, first of all, this slide was stained for elastic fibers. Okay? So you can see elastic fibers. The reason you can see this dark outline here is because it was actually stained for elastic. Okay? So what kind of a blood vessel, what kind of an artery are we looking at here? You look like you know it. No. Come on. Take a guess. Sorry? Is it the aorta? Why would you think it's the aorta? Okay. Right. So, if you were looking at the aorta, would you expect to see a lot of elastic fibers within the Tunica Media?
I'm not sure.
Well, we just looked at it. There were lots of elastic fibers in the Tunica Media. So, what we're seeing here is there is no actual elastic fibers showing up anywhere. Okay? So, aorta initially might seem like a good guess because you've got a fairly thick Tunica Media. You can see that very clearly. We've got lots of cells here. Okay? So I think this is a large vessel. Okay? But look a little deeper and say, okay, am I seeing elastic fibers? Because remember, the aorta is an elastic artery. Okay? So am I seeing elastic fibers in the Tunica Media? And in fact, we don't see any elastic fibers here. We can see that internal elastic membrane very clearly. And around the outside, you can kind of think, oh, there's some more wavy lines out there, which again would imply that there's lots of elastic fibers there. But in the Tunica Media, there's nothing. Okay? So again, initially you might think, okay, it's thick, so yeah, it's going to be a very large vessel. Aorta is the most obvious choice. But again, think about it a little bit more deeply, and you can see that maybe it's not. Okay? So again, look for more pieces of evidence and try to build a case for why you think it is what it is. Okay? So it is probably a relatively large blood vessel still. Okay? It's probably a relatively large artery. And you just, by counting the number of nuclei that you can go from one end to the other, you probably have a good 20, maybe 30 nuclei in thickness. So about 20 to 30 layers of smooth muscle. It's a fairly thick Tunica Media. So it's probably a relatively large artery. But we don't see any more elastic fibers there. So it's a muscular artery. So probably we classify it more as a medium-sized artery. Okay? Which means that this blood vessel right here would be what? Thicker in hairs. We usually pair it up such that large goes with large, small goes with small, medium goes with medium, venual goes with arterial. So if this is a medium-sized artery, what is this? A medium vein. Okay? Let's take a look at them more closely. Actually, before we do that, this structure right here, what is that? Right there.
Is that a nerve? A nerve?
Well, you're looking at fine. That's the nerve up here. This structure here. Vascular. Okay. Yeah, it might provide the vascular. It's a pretty large vessel to be a vascular. And it's not really within the adventitia of this vessel or that vessel. It's kind of on its own. What kind of blood vessel is that? A capillary. This is very low power micrograph. Capillaries are about 10 micrometers So I know it looks small on this slide, but that's just because we've really zoomed out. So it's a very low power microgram.
Is that a lymph tissue?
Hmm?
Is that maybe a lymph tissue?
Lymph tissue. We haven't talked about lymph tissues. I wouldn't be asking about that yet. Nice try though. Okay? Look at it. What can you see on it? How would you describe this vessel? Shape of the lumen. Can you tell me about the lumen? It's round. Okay, good. So right there, that kind of points you in one direction. Okay. Can you tell me anything about maybe the presence of an internal elastic membrane there?
Really get small artery.
We're looking at an artery, definitely. Whether it's smaller or not, we're going to get to that.
Well, in comparison to that.
Comparison to these guys, yeah, it's definitely much smaller. So, yeah, okay. But again, look, we do have a squiggly line here. We do have a fairly intensely staining internal elastic membrane. And then outside of that, we've got probably about 5 or 6 layers of smooth muscle. Okay. So we're looking at an artery. It's round, has a very obvious internal elastic membrane. And we do have the 5 or 6 layers of smooth muscle around it. So it's probably a small artery. Unfortunately, on this particular slide, I don't see a very obvious vein next to it. But chances are there will be a small vein somewhere nearby as well, because they do tend to run in pairs. And again, up at the very top, you can see this is what a nerve looks like. It's very poorly stained and kind of looks very weird. And we'll talk about why at the end of the semester. Let's move on. High magnification of the artery. How do I know it's the artery? Or about the tunica media? You have very many layers of smooth muscle here. So from here to here is the tunica media. The squiggly line right here is a remnant of some of those lamellae of elastic fibers that would be found within the aorta. So again, we're probably not too far away from a relatively large blood vessel, a relatively large artery. So we're branching off from one of those larger arteries. So we still have some of those remnants visible. We can see very clearly this very wavy outline right there in the lumen. This is the internal elastic membrane. At the surface of that is going to be the endothelium. And if you look carefully in a few places, you can see that there is a little bit of stuff here. That would be the subendothelial connective tissue. So it's still present, but there's very little of it visible in this case. And then all this stuff out here, part of that would be the tunica adventitia. And you can kind of make out a fairly intensely staining band out here, separating the tunica media from everything else. That would be that external elastic membrane. And then the rest of that is really connective tissue surrounding other structures as well. So the tunica adventitia becomes continuous with the connective tissue that surrounds other vessels. So don't let this fool you into thinking that this tunica adventitia is thicker than the tunica media. Okay? The tunica media really is the main feature of this particular blood vessel. And let's compare that to this right here. How do we know this is a vein? Again, the lumen's over here. You can see some white blood cells within the lumen. When we come back after reading week, you will learn how to identify what these are. You'll be able to tell me what they are. Okay. How do I know that this here is a vein? Well, let's go through the list. What do we know about the tunica intima? What can you say about the tunica intima? Try to describe it. Yeah. It's fairly thin, right? What can you tell me about this internal elastic membrane? It's not very prominent, is it? Not compared to what we just saw. Okay, so it's not a very prominent internal elastic membrane. A fairly thin tunica intima. From here to here is the tunica media. What can you tell me about it? How would you describe it? Again, remember, think back to what we just saw with the artery. What are you seeing here?
And then there's some like the layers of separated.
Okay, so the layers of smooth muscle are more separated. There's a lot more ECM there. And some of these regions here might contain more fibroblasts as well. Okay? So we're seeing a bit more ECM here, and then out here we've got the tissue. Okay? So again, we can see here that the tunica media really is not the main focus. It's not the major component of this blood vessel. And again, if we look at this at high magnification, as we just did earlier, you know that it has a much wider lumen than the artery next to it. Okay, how about this one here? So lots of adipose tissue all around, and then we've got this cluster here. Okay, what are we looking at? Okay, well let's classify them first. Arteries versus veins. Which one's which?
Artery.
So this one here? Artery. This one here? Why?
One, where they're defining artery, is a vein next to it.
Okay.
So
Well, that's
then-
because if you see an artery, there's going to be a vein next to it. Okay. Why doesn't this the artery?
Lumen size.
Sorry?
Is it
The lumen size? Okay. Is it possible that it's maybe just a larger artery? Why can't you tell me with certainty that this is a vein and not an artery?
I think this was Tunica Media.
Tunica Media thickness. What can you tell me about it?
It's not, of course, way smaller than the artery.
You've got maybe one or two layers of smooth muscle here. You just barely see it. Okay. Whereas here you have, maybe 5 layers of smooth muscle. In this case here, this slide was not stained for elastic fibers, so you can kind of make out this very faint outline, this very wavy band, which was the internal elastic membrane. It's still relatively clearly visible on this particular vessel. You can't make out an internal elastic membrane in that one. Yeah.
Now, does the smooth muscle for the artery have like lots of ECM between them?
Does it have lots of ECM between it? Well, you can see that the nuclei are relatively close together, so there's not a lot of ECM there. Okay, there is present. There's definitely going to be some reticular fibers and some elastic fibers there, but they're not very prominent here. Okay? It's very, very cellular. Okay, and right next to it over here is a nerve, just so you guys know. So over here is a nerve. I know it's not very clearly visible in this case. Okay, let's move on to capillaries. Let me finish this off. So there are 3 main types of capillaries. There are continuous capillaries, fenestrated capillaries, and sinusoids or discontinuous capillaries. So let's take a look at them individually. Now, capillaries are very, very small. They have a very small diameter. And so, in many of your slides, you're going to have a hard time really seeing them. Okay? So when you're looking at them under the microscope in the labs, you might have a really hard time picking up the actual capillaries themselves. And you're certainly going to have a really tough time differentiating between the different types. Okay? So don't worry too much about that. The discontinuous capillaries are really the only ones that you can really differentiate from the others fairly easily just because of their size. That's really all you're going to be able to tell. Okay, so, what do we know about capillaries? You guys can follow along on the handout, and I will do some more scribbling. So first of all, continuous versus fenestrated. A continuous capillary has a continuous endothelium. Okay, so you're going to have maybe a couple of endothelial cells. Making up the walls of the capillary. There is no Tunica Media or intima, etc. It's just basically an endothelium itself, and that's the capillary. With a fenestrated capillary, it's very similar. Size-wise, it's very similar. But with the fenestrated capillary, what you have are small little pores within the actual cell. I'm trying to kind of make it look like the other drawing as well, and not succeeding too much. Now, they're not going to be quite as obvious in your you're certainly not going to be able to see these pores under the light microscope. This is electron micrograph type of stuff that you'll be able to detect. Okay. So this is continuous. And this is fenestrated. Fenestra means window. So basically they have little tiny windows, little pores within them. Okay. Now, underneath the endothelium is a continuous basement membrane. So in both cases what we have is a continuous basement membrane. And so remember that basement membrane is going to be acting as a filter, as a sieve. So only some certain things are going to be able to pass through. So, what do we know about that? Continuous endothelium, continuous basement membrane. Where it's fenestrated is, well, fenestrated endothelium. But continuous basement membrane. So it'll become important when we're comparing these to sinusoids. These are found in places where you have fairly low exchange rate. Okay, so the exchange between the inside and the outside is relatively slow or relatively low. And you're only transporting very small molecules, so gases, okay, and simple sugars. So these things will be able to pass across relatively easily. The gases can simply go by diffusion, and these simple sugars will be transported by small pinocytic vesicles. So basically a little tiny vesicle pinches off, takes up something from the lumen, then migrates across the thickness of that cell, and then dumps its contents at the basement membrane. So, small molecules only. So, this is where you will see this. Muscle. You're just trying to feed the muscles. You just want to give them oxygen. That's in glucose. So you're not trying to transport very large things across. So you tend to find it there. Lungs. Again, gas exchange is the main function there. You don't really require anything about the capillary, you just want something that easily allows for diffusion to occur. Brain. Why brain? You want to exert very tight control over what passes across. Your brain is the most important organ in your body as far as your body is concerned. So it's going to very tightly regulate what is allowed to pass across that endothelium. Okay, and we'll talk about the blood-brain barrier later on this semester. And having continuous capillaries is one of the things that is a very important part of that blood-brain barrier. This requirement for the transport of very small molecules only is one of the things that drug companies have to work with. If they're trying to design a drug that's going to target a cell in the brain, they need to make sure it can actually penetrate the blood-brain barrier. So it has to be relatively small, a relatively simple molecule. Otherwise, it's not going to get in. It's not going to be able to perform its function. Okay, this here, the fenestrated capillary, allows for transport of things out of the lumen much more easily because it has these fenestra. Now, some people actually would argue that the fenestra are actually just very large pinocytic vesicles. So this cell just was transporting things so quickly that it just produced a very large pinocytic vesicle that just spans the thickness of the whole cell. Okay. Now, these fenestra are covered by a tiny little slit called a diaphragm. Which will control the passage of things across the lumen. But basically, things don't have to pass through the cell. Okay. So you can have things transported in or out, and more frequently than not, it's going to actually be into the blood vessel. Okay. So this is for transport of larger molecules. Where do you think you might need some of those? Kidneys. Great. It's a perfect example. Kidneys. In fact, in the kidneys, there's a specialized type of fenestrated capillary that actually doesn't have that diaphragm. So it doesn't even bother exerting any kind of selection on what passes across. Things just flow out. As long as they're of a certain size, they will be able to leave the blood vessel. Okay? Which basically means that larger proteins will be able to pass through, and your cells won't be able to pass through, but water and smaller ions and waste and all kinds of things will be able to pass across that endothelium very easily and very efficiently. Okay. So other places. Glands. If you have an endocrine gland, one that's producing a hormone, that hormone has to get into the bloodstream somehow. The best way to do that is through a fenestrated capillary. Your digestive tract, your small intestines. You are very rapidly absorbing material from the lumen, and that material has to be able to get into the bloodstream very easily. So again, fenestrated capillaries is what you're going to have in that region as well. So small intestines. Basically, anytime you have to have a very high exchange rate. So, I've got some drawings already pre-made for you. I don't know why I bothered drawing all this up, but it slows me down, so I'll do that as well. So, again, what you can see here is a continuous capillary versus a fenestrated capillary. You can see a continuous basement membrane around it. There's this cell type called a pericyte on this diagram, which is basically an undifferentiated cell. Okay, this cell can be used to differentiate into another endothelial cell, if, for example, there's damage to this endothelium, or it could differentiate into something like a smooth muscle fiber if this blood vessel needs to become some sort of a precapillary sphincter or something, it could become a smooth muscle. The important thing to note here is that the pericyte has the basement membrane around it as well, so it does have that epithelial sort of initial tissue type. Okay, here's an EM micrograph of a fenestrated capillary. This is taken from the pancreas. And so in the pancreas we have structures called islets of Langerhans that produce things like insulin. And that insulin has to be able to get into the bloodstream relatively easily, and so this is where we would have one of these fenestrated capillaries. Okay. In both cases of fenestrated and continuous capillaries, what we're looking at is a capillary that has about a 10 micrometer diameter. Okay. And so you can see here is a red blood cell. Again, the diameter of the red blood cell is going to be about 7 micrometers. So, in total, this capillary probably does have a diameter of about 10. But capillaries can be smaller than that. You can have capillaries, especially continuous ones, that can be as small as about 4 micrometers in diameter. Okay. This here is not capillaries. It's probably venules, just because of their size. And so what you can see here is, what I wanted you guys to see is the fact that we have an endothelium, these very flattened nuclei here. There's a branching here. This is just underneath the skin. So we're looking at a bit of a longitudinal section here through one of these blood vessels. Again, this is not a capillary because right there is a red blood cell. Another one right next to it. The diameter of this is much wider than two red blood cells next to each other. Okay, so what we're looking at here is probably a blood vessel that's probably about 20 to 25 micrometers in diameter. Okay, so it's not a capillary. It's probably a venule. Okay. But what I wanted to point out here is that there is no underlying tunica media or anything like that. All you have is just the endothelium and then just connective tissue surrounding it. Now you might notice some of these kind of very faint looking round spots within the lumen. They're not actually within the lumen. What we're looking at here is a longitudinal section through a tube. So what we're looking at here is the nucleus of the cell at the bottom of that tube. It's basically a little out of focus. But what I want you to understand is that these endothelial nuclei aren't just these little, very tiny little thin squiggles, little thin slits. They're in fact relatively round. It's just very flat. So you can think of them as kind of a pancake-shaped nucleus, where it's very flat but very thin. Okay, so that's what you're seeing here. You're seeing the nucleus here from the side, and here's what it looks like from the top. Okay? All right, so discontinuous capillaries are also known as sinusoids. And these are wider. They are larger. They have a large lumen. They tend to be used to help filter things. Okay? So the wider lumen tends to be acting to ensure that we slow down the flow of blood. Okay? So it's going to be used to slow down the flow of blood. Now, basically, how does that work? Well, if you think about it, so you've got water running through a small pipe, and then it opens up into a wider pipe. It's going to just slow down because it just spills out and fills the larger pipe. So anytime you need to do any kind of filtration, you want to slow down the flow of blood. If you want to clean out your blood, if you want to pass it through a filter of some sort, like the spleen or the liver, you're going to slow it down so that it flows down more slowly and has more time to interact with the underlying cells. The other thing about the capillaries, the sinusoids, is that the endothelium and the basement membrane are both discontinuous here. Okay, so if we were to try to draw a diagram of one of these, and we're going to try to kind of keep it relatively to scale, what you might have is one cell, here's another cell, maybe part of another cell, here's the rest of it. Here's another cell maybe. So let's just throw in some nuclei in there. And the basement membrane. Again, basement membrane is going to occur wherever you might have some of these cells. But the openings are very large and they are unobstructed. Okay? Which basically means that these pores are going to be large enough for a red blood cell to fit across and exit the bloodstream. Well, would we need this sort of thing? Well, if we're doing filtration of blood in the spleen, you want to make sure that you can allow any component in the bloodstream to leave. This way, if there's a pathogen in the bloodstream, it can also be removed, because what's underneath all these cells here are going to be components of your immune system. Okay? So there's going to be cells like macrophages, for example, that are going to be there that can take things out. Okay? And some of the cells that we'll be talking about in the next lecture will be in there as well, that will be able to remove pathogens and react to them. Okay? Another place where you might need something like this is bone marrow. How do you get red blood cells into the bloodstream to begin with? Through these. Okay, so last slide, I think I will stop here, and I will talk about the lymphatic vessels when we start talking about the lymphatic system. Okay, so we'll pick it up with the lymphatic system later on. 2 weeks from now, you guys come back from your reading week. We'll pick up with blood. The lab for that week is going to be covering this material and the material from that week's lecture. So make sure for that quiz you study both this material that we covered today, as well as the material from that lecture.